Welcome to The Explainer. Today, we're tackling what is easily the most dramatic phase of the entire cell cycle. We're talking about the M phase, or mitosis. Now, I want you to think of this not just as a simple cell division, but as a complete top-to-bottom interior renovation. We're talking entire walls coming down, genetic blueprints being tightly boxed up, sorted out, and then meticulously unpacked into a brand new space. It is a highly choreographed dance. And by the end of this explainer, you're going to see exactly how a single cell perfectly clones itself without missing a single detail.
Okay, let's dive into this. Here is our exact roadmap for today's renovation project. Number 1, the cell's renovation project. Number 2, prophase. Number 3, metaphase. Number 4, anaphase. Number 5, telophase. And finally, number 6, cytokinesis.
So let's kick things off with Section 1, the cell's renovation project. Organizing the chaos of cell division.
Look, if you're going to perfectly duplicate a cell, and by that I mean every single piece of DNA has to be copied and sorted without a single error, you have to be incredibly organized. You can't just slice a cell down the middle and hope for the best, right? So mitosis is fundamentally a stripped, two-step process to keep the chaos to a minimum. Step 1 is karyokinesis, and step 2 is cytokinesis.
Let's define that first step. The prefix karyo refers to the nucleus. So karyokinesis is literally the division of the nucleus and all the genetic material inside it into two absolutely equal parts. This is where the real heavy lifting happens. The cell has to make absolutely sure both future cells get the exact same instruction manual.
Right, so once the DNA is safely sorted, we move on to step 2, cytokinesis. Cyto refers to the cell. So this is the division of the cytoplasm and the physical cell body itself. Because, let's be honest, dividing the DNA just isn't enough. The new cell needs a physical body, it needs organelles, and it needs the machinery to actually survive and read those genetic instructions.
Moving on to Section 2, prophase, packaging the DNA, boxing up the genetic material.
Think of this phase a lot like packing up your belongings before you tear down the walls of your house. You wouldn't demolish a room with loose papers flying everywhere. So, that loose chromatin begins to untangle and tightly condense into those classic, highly visible X-shaped chromosomes. Meanwhile, those little structures on the edges, the centrioles, they start shooting out spindle fibers, kind of like microscopic grappling hooks. And crucially, to actually get to the DNA, the protective walls, meaning the nuclear envelope and the nucleolus, start to break down and disappear.
Section 3. Metaphase, the perfect alignment. The nuclear walls are completely gone.
So, as we enter metaphase, the nuclear envelope completely disintegrates, which leaves the chromosomes just floating directly in the cytoplasm. Which brings up a really fascinating practical question. If you are a biologist staring through a microscope, when is the absolute best time to observe a chromosome's shape and size?
The answer? Metaphase. This is literally the ultimate photo op. By the time the cell reaches this phase, the condensation of the DNA is 100% complete. The chromosomes are as thick and visible as they are ever going to be, making their morphology, their overall structure, perfectly clear for observation.
And this brilliantly illustrates what those spindle fibers are actually doing. Notice how they reach out and lock onto these specific protein structures called kinetochores right at the center of the chromosomes. Once they're attached from both sides, they play this intense microscopic game of tug-of-war until all the chromosomes are perfectly aligned right down the middle, creating what we call the metaphasic plate.
Section 4, anaphase, the great divide, pulling the blueprint apart.
Okay, so this is the shortest phase, but arguably the most intense. The tension peaks, and those identical sister chromatids are forcefully ripped apart right down the middle at the centromere. The spindle fibers basically just reel them in toward opposite poles. And if you look closely at how the chromosome arms trail behind the centromeres as they're dragged through the fluid, it creates these really distinct shapes. You get these tiny V's, L's, J's, or I's just flying through the cell.
Let's actually put some numbers to this to understand what scientists call equational division. We'll use human cells as an example. So, 46 chromosomes line up in the middle. When they split, 46 chromatids get pulled to the top pole and 46 get pulled to the bottom. The moment they separate, they are officially considered individual daughter chromosomes. The total number stays exactly the same.
Section 5, telophase, rebuilding the nucleus, unpacking in the new space.
You can kind of think of telophase as essentially the exact opposite of prophase. The moving is finally done, and now it's time to unpack.
Those tight individual chromosomes loosen up and decondense right back into that messy, tangled chromatin. At the exact same time, the cell starts rebuilding the walls. Notice those dashed lines reforming? That's the nuclear envelope reappearing around both sets of genetic material. So by the end of this phase, you have one cell body, but two fully formed, completely genetically identical nuclei.
Section 6. Cytokinesis. Splitting the room. Dividing the physical cell body.
Okay, so the DNA is perfectly divided, the nuclei are rebuilt, but we still have one giant room. We have to divide the actual physical space. Let's move to and see how this builds, because plant and animal cells handle this drastically differently. For animal cells, they have a soft, flexible membrane, so they simply squeeze. A cleavage furrow pinches from the outside in until the cell just snaps into two. But plant cells, they have rigid cell walls. They literally can't pinch. So instead, they build a brand new wall, a cell plate, right down the middle, working from the inside out.
Now here's a wild thought. What happens if a cell finishes step 1, but completely fails to do step 2? If a cell successfully divides its nucleus in karyokinesis, but fails to divide its body in cytokinesis, you end up with a condition called a syncytium. It's a single, giant cell, absolutely packed with multiple nuclei. It's this fascinating biological glitch that really reminds us why this strict, two-step ordered process is so incredibly vital.
And that brings us to a pretty provocative final thought. We've just seen that mitosis is a flawless equatorial division. 46 chromosomes go in, 46 go out. It perfectly clones a cell, ensuring no information is lost and nothing is changed. But if our cells are constantly just cloning themselves, how do genetic variations, evolution, or unique traits ever occur in a living organism? Where does the diversity actually come from? Well, that requires a completely different type of biological renovation project called meiosis, and we are definitely going to save that fascinating explainer for another time. Thanks so much for joining me today, and keep exploring.